So our next speaker is Miss Christy Hudak. Christy has been a research associate in the Right Well Ecology Program at the Center for Coastal Studies for 10 years. After obtaining her master's degree in marine science from Nova Southeastern University in Florida, Christy worked at the Florida Fish and Wildlife Commission, heading the manatee program at the Tequesta Field Station. From the sunny shore of Florida, Christy then brought her experience in marine mammal necropsis and rescues, animal tracking, aerial surveys, and polymer identification, amongst the many others, to the cool shores of Cape Cod. And currently, she focuses on the food resource of North Atlantic right whales and microplastic research. Welcome, Christy, and the floor is yours.
Thank you so much for that great introduction. Hello! Today I will be presenting some of our results pertaining to the ingestion of plastics in Great Shearwaters. My colleagues and I focused on the plastic ingestion and bycatch demographics of Great Shearwaters from the Gulf of Maine, since the U.S. coast and Gulf of Maine in general are more poorly described in terms of plastic ingestion in marine biota compared to other regions. Despite the high density of human populations and the potential plastic sources along this coastal margin. However, we were given the opportunity to expand the project to include other habitats of the Great Shearwaters along their migratory route. Since the 1950s, globally monitored seabird populations have declined by 70%. Regionally, large-scale Great Shearwaters mortality events have likely increased in frequency and magnitude along the U.S. Atlantic seaboard due to uncertain causes. Plastic pollution, fisheries bycatch, and changing foraging conditions have been cited as possible causes in observed seabird declines. The ingestion of plastic is not rare. It is common throughout all trophic levels, from plankton to the top predators, with plastics found in a wide spectrum of sizes, shapes, and types of plastic materials, depending upon the species. Unfortunately, seabirds are considered excellent indicators of plastic pollution, with procellariiforms such as Great Shearwaters, albatross, and storm petrels being the most prone to plastic ingestion. Since the 1960s, it is estimated 59% of seabird species examined and ingested plastic, with an average of 29% of those individuals having plastic in their GI tract. Scientists have predicted that by 2050, almost 100% of the seabird species will have ingested some form of plastic debris. It is not limited to just seabirds. The percentage of species ingesting plastics in numerous other animal groups is increasing as well. Our study focused on the accumulation of ingested plastics in Great Shearwaters. Seabirds offer a sampling strategy to understand stressors contributing to possible declines, as their assessed individual health and population condition can be utilized as an ecosystem indicator, reflecting biological, physical, and chemical conditions across spatial and temporal scales. They're a long-lived pelagic bird and a trans-equatorial migrant, inhabiting the Gulf of Maine waters during the non-breeding season in the summer, and traveling to the breeding grounds located on the St. Helena Island group, which is over here, and the Falkland Islands in the South Atlantic. Tracking and necropsy data also suggests age-specific segregation of their non-breeding range, with preferential use of the southwest Gulf of Maine and Massachusetts Bay by the juveniles. The Great Shearwaters' primary food resource includes squid, crustaceans, and small forage fish, such as sandlance. They typically feed by pursuit plunging or by surface seizing behaviors, with most foraging activities occurring within the top 10 meters of the water column. Now, the average dive depth is approximately 2 to 3 meters. Even though a large portion of plastic debris may be dense enough to sink or become biofouled and sink to deeper waters or the ocean bottom, it is still estimated the ocean surface layer contains trillions of plastic particles in various sizes and composition, directly in the path of the foraging birds. In addition, Trophogenialists and surface-seizing birds reliant on crustaceans diets have been found the most vulnerable to plastic ingestion. What we know is that monitoring plastic ingestion in these species is valuable due to its co-location with key marine megafauna and its representation of divergent Atlantic habitats, including developed coastal margins within the Gulf of Maine and pelagic regions in the South Atlantic. While knowing the types of polymers is valuable in aiding in the identification of potential plastic sources, for instance, polyethylene products versus polystyrene products, the polymer composition of ingested plastic is related to spatial patterns of plastic abundance rather than a preference for ingestion of any one polymer type. Example, a study done on an ecologically similar procephaliform, northern fulmar, demonstrated seabird plastic ingestion scales to plastic abundance in surface waters. In essence, the more plastic debris that is in the area, the more plastics they will ingest. Our goals were to evaluate any trends in ingested plastics over time and compare the plastic categories and polymer types between locations and time periods. Our samples came from birds caught in fishery gill nets or washed up on shore between 2010 and 2019 from 3 locations: Massachusetts Bay, southeastern shores of North Carolina, Gough Island in the South Atlantic. We performed standard necropsies on 175 birds and analyzed the ventriculus portion of the GI tract for plastics. This is the mid portion of the stomach in birds, so it's also known as the gizzards. Plastic material was sorted by established qualitative categories, either industrial or user plastic, and further divided into 5 descriptive subcategories. This includes sheet-like plastics, which includes bags and film. There are thread-like plastics, which includes microfibers, rope, and line. You have the foam category, which is polystyrene. The fragment category pertains to unidentifiable hard plastics, and then all the rest goes into the other category, which is balloons, rubber, or melted plastic. Each piece was weighed and measured, and then classified into plastic size categories, either micro, meso, or macroplastics. Using an FTIR spectrometer, we categorized the pieces into 23 polymer types. We teased out polymers with additives or breaking down polyethylene into low or high density, if possible, based on spectral interpretation. The reason I say if possible is due to the difficulty in identifying between the low, medium, or high density polyethylene because the spectra are so similar and the degradation of the plastics in the Great Shearwater gizzards changed the polymer spectrum enough to make the identification difficult. For this presentation, I minimized the polymer categories down to 13 general polymers. Out of the 1,638 pieces collected, there were a total of 1,570 plastic pieces. Other materials that were found in the gizzards include rocks, vegetation, natural fibers, and biologicals. As expected, based on previous studies done on the polymer composition of plastics in the surface waters and general areas, the majority of plastic material was composed of polyethylene at 75%, followed by the mixture of polypropylene and polyethylene at 13%, and polypropylene at 7.5%. The rest of the categories made up the other approximately 4%. Grouping the polymers even further to 7 categories, we examined the polymer composition by year and age class, and age class included breeding, non-breeding adults, and juveniles, and by location, as you see up here. As you can see, polyethylene had the highest percentages based on the years, the location, and the age class. One interesting note, the mature breeding birds on Gough Island had a higher percentage of polypropylene polyethylene mix than compared to any of the locations or life stages. Next, we compared the size of fragments between habitats. We found mature birds from Gough Island contain the largest fragments, significantly larger compared to items found in individuals from Massachusetts Bay or North Carolina. In terms of average number of pieces, North Carolina had the highest average with 12 pieces per bird compared to 8.5 pieces per bird in Massachusetts Bay, or the average of 9.6 pieces per bird for Gough Island. Finally, we focused on 5 descriptive categories: the fragments, the sheet-like, the thread-like, foam, and other. And this graph shows the percent of plastic category through the years and by location. As you can see, fragments comprise the majority of the pieces, both by year and by location. And an interesting note, in 2018, a higher number of other category, higher percentage, which contained balloon-type material found in the gizzards. In summary, polyethylene and polypropylene ethylene mix were the most prominent plastics in Great Shearwater gizzards, reflective to their foraging habitats, since plastic pieces within coastal and pelagic surface environments are predominantly composed of polyethylene and polypropylene, which are the 2 of the most highly produced olefin plastics used in an array of single-use consumer and industrial products. There were significantly different polymer signatures and sizes between the North Atlantic and the South Atlantic birds, suggesting plastics turnover within the GI tract corresponds with migratory phase, allowing the Great Shearwaters to reflect their ambient environment during an assumed acclimation period. Juveniles and non-breeding adults had higher frequency rate of accumulated plastics than mature adults in their breeding phase. And finally, fragments were the dominant plastic item ingested across all stages, from juveniles to breeding adults, all habitats studied, including Massachusetts Bay, North Carolina, and Gough Island, and through the years of 2010 and 2019. I want to thank all our collaborators and supporters of the Great Shearwater project, because without their expertise and financial support, this project would not have been possible. Thank you.